So hello students and welcome back to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the DNA topology, twist and wreath. So what is this twist? What are wreath? How twist and wreath are being interrelated with respect to each other? What is a linking number? And how does the enzyme topoisomerase work? In what manner the enzyme topoisomerase work? How does the action of topoisomerase regulate twist and wreath in the structure of a double-stranded DNA molecule? Now, the entire procedure we must understand with the understanding of DNA topology, twist and wreath lecture. Okay, so now please have a view of the double-stranded DNA molecule and the supercoiling associated with this DNA molecule.
Topology describes the coiling of a molecule of DNA. When you have completed this exercise, you should understand why the linking number for a molecule of covalently closed circular DNA, or cccDNA, is constant. Understand the twist component of the linking number. Understand the writhe component of the linking number. And understand how cells utilize supercoiling.
So, in this particular lecture, we will be paying our attention in order to understand the concept of linking number. What is this linking number? For a molecule of cccDNA, which is referred to as circular covalently closed DNA molecule. This DNA molecule, which you can visualize over your screen, is a cccDNA molecule. We must understand the twist component of the linking number. We must also do understand the writhe component of the linking number, as well as understand how do cells utilize supercoiling. What is this supercoiling and how do cells uses this supercoiling? And we must understand that what happens in the case of this dsDNA, which is also referred to as cccDNA, that is circular covalently closed DNA molecule. So please look here.
Note: many of the DNA structures shown in this exercise are meant to serve as models to aid in the understanding of twist and writhe. They do not represent actual structures found in living cells.
Okay. So many of the
Each of
structures
the chromosomes of
that
eukaryotes
were being given in this particular exercise, they just represent the different types of structures which are being associated with the cell genomic DNA. Now, the genomic DNA in the case of cells, when you talk about the genomic DNA in the case of cell can be of two different types. It can be linear, it can be circular. It can be linear, as is the chromosomal DNA. Linear is chromosomal DNA. Linear is chromosomal DNA. Chromosomal DNA is a linear form of a DNA molecule that is present. This is referred to as the chromosomal DNA. Okay. This chromosomal DNA is also 5 prime to 3 prime or 3 prime to 5 prime, 5 prime to 3 prime. Okay, and it is bounded at its ends with the help of certain proteins. Its ends are never let free. They are being bounded with the help of certain proteins. The proteins that binds towards its end are referred to as telomerase binding protein. ये जो प्रोटीन्स इसके किनारे पे बाउंडेड होती हैं, इन प्रोटीन्स का नाम होता है टीलोमेरेज बाइंडिंग प्रोटीन. द प्रोटीन्स which are being bounded at its ends is referred to as the telomerase binding protein, also referred to as TBP. 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 Telomerase binding protein. Okay, so these telomerase binding proteins are going to bind specifically at the telomeric region. They are going to bind specifically at this particular region, which is referred to as telomeric binding proteins at the telomerase region. Now, the next point, or the genomic DNA can be even circular. Instead of being a linear DNA, it can be even circular. circular chromosomal DNA. When you talk about circular chromosomal DNA, circular chromosomal DNA is present in the case of bacterial cells. In the case of bacterial cells, you always know the chromosomal DNA which is present in the case of bacteria. The DNA which is present in the case of bacteria is always circular and covalently closed. This is the type of DNA which is present in the case of bacteria, but this DNA is CCC DNA. It is circular covalently closed DNA. As is the case of plasmid DNA, you can also observe the plasmid DNA that is associated with bacterial cells. Okay, this is also a CCC DNA molecule. So here the DNA is called as CCC DNA. So both linear DNA, which is bounded at its ends, and circular covalently closed DNA molecule are referred to as topologically constrained DNA molecule. Both the linear as well as the circular dsDNA molecule are said to be the topologically constrained DNA molecule, are said to be the topologically constrained DNA molecules, as these cannot undergo any change in their linking number without the help of topoisomerases. Okay, so they cannot alter their own linking number without the help of topoisomerases. That is why both of these DNA, whether you talk about the linear chromosomal DNA molecule or whether you are talking about the circular chromosomal DNA molecule, both of these are said to be topologically constrained DNA molecule. Now please look at the animation.
Karyotes is comprised of a single linear DNA molecule. Because their ends are free, linear DNA molecules can freely rotate to change the number of times the two chains of the double helix coil about one another.
So here you find out that because when the ends of the DNA are free, they can freely rotate around each other and can get denatured spontaneously. Here what you have find it out is that spontaneous degradation of the DNA or spontaneous denaturation of the DNA is going to take place if the ends are free. But if the ends are bounded, then the spontaneous degradation or denaturation of the DNA will not take place. So please note it down here that when ends of the linear DNA molecules are free, these can effectively undergo denaturation. These can effectively undergo denaturation and separation. So only the linear DNA bound at its ends, as well as the circular, as well as the CCC DNA, are sufficiently stable in a cell. So only both of these DNA molecules are stable in a cell, whereas the other DNA molecules which are free, whose ends are free, are never going to be stable. Okay, clearing all the drawings and moving forward.
However, DNA molecules can also be circular. Examples of circular DNA molecules include most bacterial chromosomes, some viral chromosomes, and small bacterial genetic elements called plasmids. For such covalently closed circular DNA, the absolute number of times the chains coil about one another cannot change. These DNA molecules are said to be topologically constrained.
Okay, so you can look here. This is what I have explained it to you. A DNA molecule whose ends cannot be separated is said to be topologically constrained. When this particular DNA molecule was present in its linear manner, then also the number of turns that were associated with this DNA were 5. Now also the number of turns, even when it has been stretched, remains 5 only. So the number of turns are not going to be varied in such type of a double stranded DNA molecule. So what you found here is that the number of turns have remained constant. Okay, so this is the property of a DNA molecule that is said to be topologically constrained. Okay, moving forward, such type of DNA are the plasmid DNA as well as the circular genomic DNA, which is found to be associated with the nucleus of the eukaryotic cell.
Even the linear DNA molecules of eukaryotic chromosomes can be topologically constrained because of their interactions with other cellular components.
So, in fact, the linear DNA molecules can also be topologically constrained. So, you must not see that only the circular DNA molecule is topologically constrained. Even the linear DNA molecule is topologically constrained because of the presence of telomerase binding protein associated with the ends of the linear chromosome, that is the telomere.
Since each of the two strands of cccDNA are covalently linked circular chains, and because they coil around one another, the strands cannot be separated from each other without breaking a covalent bond and opening the circle of one of the strands. The two strands could be separated if they were able to magically pass through each other. The number of times one strand would have to be passed through the other strand in order for the two strands to be entirely separated from each other is called the linking number.
Now, because you can find here that
The linking number
the DNA molecules were coiled with respect to each other, because you can find that the two DNA molecules were coiled with respect to each other. So what we were able to determine is that both the DNA molecules cannot get simultaneously or spontaneously separated from each other by their own activity. So these cannot be separated by themselves from each other. For the separation of such type of DNA molecule, we require a class of enzyme, and that is referred to as topoisomerase, because the two different types of DNA which are being wrapped over each other lead to the formation of a topologically constrained DNA molecule. As it leads to the formation of topology constrained DNA molecule, so these can be separated with regard to each other by the action of topoisomerase, which could cross one strand of the DNA over the other. Okay, the linking number describes the topology of a DNA molecule. So you are going to describe the topology of the DNA molecule with the help of linking number. Now, how to define the linking number? The linking number refers to the number of times one DNA the number of times one DNA strand has to be passed through the other DNA strand in order for the two strands to be entirely separated from each other. The number of times a strand of DNA must be separated over the other strand in order that the two strands of the DNA can be effectively separated from each other is called as linking number. Are you able to understand it? If not, please do comment.
is always an integer. The linking number is an invariant topological property of cccDNA. No matter how much
See, linking number is a invariant property of the circular covalently closed DNA molecule. Every topologically constrained molecule has a particular linking number. When the linking number is present in relaxed state, then it is called as standard linking number. Now, what is this standard linking number? What is the relaxed linking number? What is the constrained linking number? All of that has to be seen. We are going to visualize it through a PowerPoint animation. Okay.
How much a cccDNA molecule is distorted, the linking number remains constant. The linking number is the sum of two geometric components called twist and writhe.
So you can find that the linking number for any DNA molecule is actually a sum of two geometrical component. One is called as the twist, another one is called as the writhe. So the twist and the writhe are combined together in order to create a linking number. Now, what is this linking number and how this linking number is being produced? So that we have to see. Next point.
Twist is simply the number of helical turns of one strand about the other. In other words, the twist number is equal to the number of times one strand crosses in front of the other strand. If cccDNA is lying flat in a plane, the linking number is equal to the twisting number.
Okay, so this is important. If the circular covalently closed DNA molecule is lying in a flat plane, then what we are going to find is that the twist number is going to be equivalent to the linking number. So the linking number and the twist number are going to be the same when the when the circular covalently closed double stranded DNA molecule is lying over a plane. When the cccDNA is lying over a plane, then the twist number is equivalent to the linking number of the DNA molecule. Look forward. Here, the width is equivalent to 0. What is a twist number? Please do listen very carefully. Number of times one DNA strand is wrapped over another DNA strand is called as twist number. What would be width? The number of times a DNA helix would be wrapped over another DNA helix will be referred to as width. What would be the linking number? The number of times one DNA strand has to be rotated over the other DNA strand so that the two DNA strands become completely separated is referred to as linking number. Looking at the width component of the linking number.
If the twists form a right-handed helix, as in DNA under normal conditions, they are given a positive twist number. To determine if a helix is right-handed, think of it as a screw. You would have to twist a right-handed screw to the right, clockwise, to screw it into a board. Also, in a right-handed helix, the strand in front at a cross point is pointed up to the right.
Now, please do listen here. Now, this is the first type of the coiling that is present in the case of DNA. You can find that in such type of coiling that is present in the DNA molecule, the twist number is considered to be positive and the linking number is also considered to be positive. Here the twist number is positive. Here the linking number is also positive in nature. Twist number is greater than 0 for right-handed DNA helix in nature. It is greater than 0 if you are going to form a positive twist number. If you are going to form a positive twist number, then such type of DNA molecule will be referred to as the positively supercoiled DNA molecule because they are having plus 5 linking number that can be visualized here. Here you can just find what is twist. The number of times one strand is going to cross over the other strand is called as twist. You can just remember this with the direction in which the screw was moving. How the screw was moving, in what manner the screw was moving can be used to predict what is twist. Next point.
If the twists form a left-handed helix, as can occur under special circumstances, they are given a negative twist number. You would have to twist a left-handed screw to the left counterclockwise to screw it into a board. Also, in a left-handed helix, the strand in front at a cross point is pointed up to the left.
So here you can find that this is the left-handed helix and this is the right-handed helix. When you are going to rotate the DNA molecule in the right-handed direction, then it is called as right-handed helix. When you are going to rotate the DNA molecule in the left-handed direction, then it is referred to as left-handed helix. On the basis of the rotation of the DNA either towards right or towards left, you are going to classify the DNA helix into a right-handed helix or into a left-handed helix. Okay, next point.
However, cccDNA does not generally lie in a flat plane. Instead, the DNA double helix usually crosses over itself in three-dimensional space, often repeatedly. This torsional distortion is called writhe.
Okay, so there is the torsional distortion that is going to be produced in the case of a CCC DNA molecule. Often the CCC DNA molecule, this is a double-stranded DNA molecule. This double-stranded DNA molecule is not lying in a particular plane, but it is coiled over itself. And this structure which is being produced is commonly referred to as the rith. Any doubt? If you have any, let me just make the structure. See this. This is the double-stranded DNA molecule. I'm trying to make a double-stranded DNA molecule. This is a double-stranded DNA molecule running in this particular manner. Actually, this is a chromosomal DNA, which is CCC, which is closed circular DNA molecule, which is present. See this? Please look here. Just a second. This is the DNA molecule that is present. Now making another strand of this DNA molecule with another color. Okay, this is the other strand of the DNA molecule. This is the other strand of the DNA molecule. Now you can find that the two-stranded, double-stranded DNA molecule is actually packed over itself. Okay, then it had led to the structure that is referred to as wreath. Because the double-stranded DNA is packed over itself, that is why the structure is referred to as wreath. If you look here, if you look here, let this be a double-stranded DNA molecule be like this. If this is the double-stranded DNA molecule, okay, and this is the actual double-stranded DNA molecule, which must have been like that, then here you find that the total amount of wreath in this case was equivalent to zero. This wreath was equivalent to zero. The twist which was present, you just count the number of twists. How many number of times the DNA strand is crossing over the other? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, something like that. You can count it out. Okay. How can you count the twist? Just just by rotating a double strand, just by rotating a single strand over the other. Then how many rotations are needed of 180 degree so that the two strands of the DNA becomes completely separated. Now supposingly 8 rotations are needed, then the twist becomes equivalent to 8 and the width becomes equivalent to 0. In this case, the width is 0, but here you can find the value of width is equivalent to 1, 2, 3. 3. Now, when you take a wire, a coiled wire over your hand and try to overwind the coiled wire, then what you will find, the wiring will begin to coil over itself, and this is the condition that is going to be width. When width is going to be increased, the twist automatically decreases. Twist automatically decreases because in all condition because in all conditions linking number remains constant. Lk remains constant. Any doubt? Any problem? Any doubt? Any problem? You just provide in the chat box, in the comment box. Now moving forward.
Writhe can take two forms. One form is interwound or plectonemic writhe. In interwound writhe, the DNA is wound around itself, forming a helix. Most CCC DNA molecules exhibit interwound writhe.
Now, what do you mean by interwound wreath? Actually, the wreath can also be of two different types. One, it could be an interwound wreath, or another, it could be a spiral wreath. Now, what do you mean by interwound wreath? And how does interwound wreath or the plectonemic wreath is being produced? So that we must visualize, okay, through this animation. Moving forward with this animation.
The writhing number is the total number of writhes in a molecule of DNA. If right-handed DNA forms a right-handed interwound helix, the DNA must twist to accommodate the writhe. Therefore, right-handed interwound turns are assigned negative writhing numbers. Note that, although the linking number must be an integer, the twist and writhing numbers can have fractional values.
Now, although the number must be an integer, but here the twist and the rising number can have different values. You can see that the twist number should be greater than 0 and should be present in positive. The width number should be present in negative. Okay, next point. So width value is always less than 0 for right-handed interwound turns.
If right-handed DNA writhes to form a left-handed interwound helix, the twist number of the DNA decreases. Therefore, left-handed interwound turns are assigned positive writhing numbers.
Now, this is important. This is very, very important. See, when width number is going to decrease for right-handed interwound turns, when width number is going to increase for left-handed interwound turns. Now, for a right-handed DNA molecule, if you are going to make a width, the width will always becomes left-handed. So the width value increases and the twist number decreases. For the right-handed DNA molecule, the width will become in left-handed direction. Width will come out to be in left-handed direction. Why? You take a wire and try to rotate that wire. Okay. While rotating that wire, you will find that the width, the coiling of the wire double stranded over itself will always be present in left-handed direction. And so the value of width is always positive for right-handed supercoiling. So when the value of width is always greater than 0, then it is referred to as left-handed interwound turn. The width value is going to be increased and the twist value is going to be decreased. Okay. When the width value is going to be decreased for right-handed interwound turn, that means now the DNA is relaxing. The twist number is going to be increased. Please look here.
The other form of writhe is spiral or toroidal writhe. In spiral writhe, the DNA winds as if around a cylinder. Spiral writhe often occurs when DNA molecules wrap around proteins.
Now, spiral wreath is another type of wreath that is going to take place. Spiral wreath is a type of wreath that takes place around a particular protein molecule. You find a protein molecule around this particular protein molecule, DNA is wrapped, and such type of wrapping leads to the production of a wreath that is commonly referred to as the spiral wreath, also referred to as the toroidal wreath. Now, in this type of toroidal wreath, everything is going to be in opposite direction. Why? Because DNA is going to bind with a protein molecule. DNA itself binds over a protein molecule because DNA has an affinity to bind with proteins. DNA being negatively charged will bind with protein, and instead of relaxing itself will make itself much more super coiled, so that the entire values will be reversed. In the case of spiral wreath, the width is going to be increased for right-handed DNA, right-handed DNA molecules, and width is going to be decreased for left-handed interwound turns.
If right-handed DNA writhes to form a right-handed spiral coil, the twist number decreases. Therefore, right-handed spiral turns are assigned positive writhing numbers. Note that this is the opposite.
Now, please look here. This is about the right-handed DNA molecule, which were making interwound width. Interwound widths are always being produced by DNA itself, whereas spiral wreath are actually being produced by the DNA binding over the protein molecules. When the DNA is going to bind over the protein molecules, its entire conformations are going to be altered because there is a protein molecule. So instead of relaxing itself, it will make itself much more super coiled. The twist number, which was decreasing, is going to be increased.
Opposite of right-handed interwound turns.
See this, it is increasing. Because here the DNA is winding in opposite direction. That is why instead of decreasing the writhing number, this writhing number is going to be increased.
If right-handed DNA writhes to form a left-handed spiral coil, the DNA must twist to accommodate the writhe. Therefore, left-handed spiral turns are assigned negative writhing numbers. Again, this is the opposite of left-handed interwound turns. Under normal physiological conditions, the average structure of DNA has about 10.5 base pairs per turn of the helix. Therefore, the linking number of fully relaxed DNA can be calculated by dividing the total number of base pairs by 10.5 to find the number of twists. This linking number is assigned the symbol LK0.
Now, how to calculate the linking number? As a matter of convention, you know that through mica experiment, the number of base pair per helix of the DNA is 10.4 inside the cell. Outside, it has been taken to be 10.5 in order to facilitate the calculations. What was this mica experiment? Why mica experiment was a simple experiment in which a sheet of mica was taken. Mica, the sun mica that you use in your homes, that sheet was being taken. Over that particular sheet, DNA was being placed. So I'm placing the DNA over a sheet, over a mica. Okay. Once the DNA was being placed over this mica, the DNA was subjected to restriction endonuclease digestion. Okay. So what was being done? DNA is subjected to mica experiment. I'm just telling you about an experiment that is referred to as mica experiment. This mica experiment it predicted that it predicted that 10.5 number of base pair per helix are present. 10.5 number of base pair per helix are present within the structure of DNA. Are present within the structure of DNA. The structure of dsDNA. How was the experiment being done? The DNA was being taken, attached to the mica, okay, subjected to degradation, subjected to Endonuclease cleavage. DNA subjected to endonuclease cleavage. DNA subjected to endonuclease cleavage. Endonuclease cleavage. Endonucleases are the enzymes that will bind to a particular site, and they will be the one that would be cleaving the DNA molecules at particular nucleotide sequences. Okay, so the endonucleases are going to bind to the DNA molecule and they are going to cleave the DNA molecule. But where the endonuclease will bind? The endonuclease can only bind to the locations where the DNA will be free. This is endonuclease. Endonuclease. Now, where the DNA will be free? The DNA will be free towards the end of a particular helix. After one helix, the DNA is going to be free. Okay. The after restriction digestion, the restricted digested fragments were subjected to gel electrophoresis. After restriction digestion, after restriction digestion, DNA was subjected to electrophoresis. DNA was subjected to electrophoresis. DNA fragments were subjected to electrophoresis. To electrophoresis. When DNA segments were subjected to electrophoresis, the electrophoresis was being completed. The electrophoresis was being performed. Now I'm going to just show you how the electrophoresis was being performed, and then the different DNA strands were being produced. Okay, I'm just going to make certain DNA strands. One strand of DNA, another strand of DNA. This is the DNA strands which were being produced. Each DNA strand which was being produced was present after a repetition of 10.4 number of base pair. Each strand was present after a repetition of 10.4 number of base pair. That is why the DNA was said to have 10.4 number of base pair per helix. As each of the fragment obtained was was different from the other by 10.5 base pair. So, so the DNA molecule was considered to be of 10.5 base pair per helix. Per helix. Full stop. How it was being done? DNA fragments produced were DNA fragments produced were 10.5, approximately in between 10 and 11, that is 10. Then it was 21 base pair. Then it was in the multiple of 10.5, they were there. It was 32 base pair, and so on. And so this is how the DNA molecule was present. That is why it was considered that the length of the DNA is equivalent to 10.5 number of base pair per helix. Okay. So now, when the entire DNA structure, you are going to look at the entire DNA structure. The entire DNA structure is of 378 base pair. Then the standard linking number for this DNA is equals to 378 divided by 10.5 equals to 36. This 36 will now be referred to as the standard linking number. This is LK naught, equal to 36. This would be referred to as the standard linking number. Standard linking number. Okay, this is referred to as the standard linking number. So what is going to happen? This is very important. Please, I'm going to conclude everything within a single line. Just look here. If you are going to find that linking number becomes equal to LK naught, hence DNA is said to be relaxed. DNA is relaxed. Okay. When you look here, LK is less than LK naught. DNA is said to be negatively supercoiled. Negatively supercoiled. Supercoiled. Now, when you are going to look here, LK is greater than LK naught. Okay. LK is greater than LK naught. Then the DNA is said to be positively supercoiled. Please note this particular relationship in between the different LK naught values. This relationship is of the most important type. LK less than LK naught, negatively supercoiled. LK greater than LK naught, positively supercoiled. So if the number of linking number increases to 37, positive supercoiled. If the number of linking number decreases from 36 to 35, DNA is said to be negatively supercoiled. Is this clear to you or not? Please, please, now you're free to ask the queries. You can ask me as many number of queries as you want to. Moving forward to the next slide. Please take a pic of this particular slide as well as this particular formula. Moving to the next slide.
If writhes are
Deleting all the annotations. Moving forward.
introduced to the DNA without changing the number of twists, the linking number changes. The difference between the actual linking number and LK naught is the linking difference. If the linking difference is
Now you can see if the new linking number is 32, that means the new linking number is less than the standard linking number. The DNA is said to be negatively supercoiled. This is called as linking difference. The linking number of the present DNA minus the linking number of the standard DNA. If it comes out to be negative, the DNA is negatively supercoiled. When you talk about negative supercoiling, that means the DNA is quite relaxed. Okay. When the value of LK is greater than LK naught, for example, 37, 38, 39, then this value comes out to be positive because this is equal to LK minus LK naught. Okay. So this value is actually LK, the linking number of the DNA molecule under investigation, minus LK naught, the standard linking number. Okay. Now move forward.
significantly less than 0, the DNA is negatively supercoiled. If the linking difference is significantly greater than 0, the DNA is positively supercoiled.
Okay, so this is the value. If linking number is less than 0, the DNA is negatively supercoiled. If the linking number is greater than 0, the DNA is said to be positively supercoiled. This is referred to as supercoiled DNA. DNA for which linking number is significantly different from 0. If this linking number is in negative, it is said to be negatively supercoiled. If this linking number is in positive, it is said to be positively supercoiled. See, if the linking difference is significantly less than 0, the DNA is said to be negatively supercoiled. If the linking difference is significantly greater than 0, the DNA is positively supercoiled. Thank you.
cccDNA from both bacteria and eukaryotes are usually negatively supercoiled. Negative
Now, this is very important. Now, can you tell me when the bacteria, when the negative supercoiling, and where will be the positive supercoiling present? When you talk about cccDNA, when you talk about circular covalently closed DNA molecule, in the case of a bacterial cell, you talk about the supercoiling being negative. When you talk about a circular covalently closed DNA molecule, in the case of a bacteria surviving in thermonuclear vent, the supercoiling will be positive.
supercoils can be thought of as a store of free energy that aids in strand separation for processes such as DNA replication and transcription. When right-handed interwound turns in the DNA are relaxed, the twist number decreases and the DNA unwinds. Ask yourself, under what conditions might an organism need positively supercoiled DNA?
Now, first and the foremost thing, the DNA is said to be negatively supercoiled, means that the DNA could be effectively separated and the two strands can be effectively separated from each other. Now, the second point, just a second. The second point, when will you say that the DNA might have a positive supercoiling? When the DNA is present in a in a organism that is going to survive in thermophilic environment, then to facilitate the DNA supercoiling, positive supercoiling is present. Now, I'm just going to write the answer. Please look here. In order to facilitate the survival of an organism in harsh environmental condition, I'm just writing the answer here. In order to facilitate the survival of an organism in the harsh environmental condition such as thermonuclear vent or in thermophilic bacterial cells, the DNA is made stabilized with the help of positive supercoiling. This is why positive supercoiling is present. Positive supercoiling occurs in the case of Thermophilus aquaticus. Thermophilus aquaticus. Yes, you are absolutely correct. This is the same organism from which the Taq DNA polymerase is being obtained, as well as the Pyrococcus furiosus, as well as the Pyro COCCUS furiosus. Okay, so these are the organisms that have such type of supercoiling present within themselves. Any doubt? Moving forward, clearing all the drawing and moving forward.
DNA molecules with the same sequence and length but different linking numbers are called DNA topoisomers, since they differ only in topology. Topoisomerases are enzymes that change the linking number of a DNA molecule.
Now, DNA molecules that differs in their topology are referred to as DNA topoisomers. Topoisomerases have the ability to convert one form of the DNA into another by altering or changing their linking numbers.
There are two types of topoisomerases. Type 1 topoisomerases increase linking numbers in steps of 1 by introducing an
See, there are two different types of topoisomerases. One is called as type 1 topoisomerases, another one is going to be the type 2 topoisomerases. So type 1 topoisomerases, when you are going to talk about, they are going to increase the linking number by 1. That means they are going to add a positive supercoil in the structure of the DNA. How? Please look here.
Additional twist. Type 1 topoisomerases relax negative supercoils.
They introduce positive supercoiling or they relax negative supercoiling. So when our DNA has to relax, we use type 1 topoisomerases. Our DNA is negatively supercoiled, so it has to be relaxed by type 1 topoisomerases, which will introduce a positive supercoil.
Type 2 topoisomerases change linking numbers in steps of 2. Type 2 topoisomerases require energy from ATP hydrolysis.
Now, type 2 topoisomerases is another type of topoisomerase. This type 2 topoisomerases alters the linking number by a value of 2. It uses ATP hydrolysis in order to cleave a double-stranded DNA molecule and allow the other strand to pass through the first strand.
For their action. By changing the handedness of turns, type 2 topoisomerases change the topology of DNA molecules.
So the basic difference, type 1 topoisomerases act over negative supercoiling, induces positive supercoiling. Type 2 topoisomerases act over positive supercoiling, introduce negative supercoiling. Type 1 topoisomerases increases linking number by 1. Type 2 topoisomerases decreases linking number by 2. Type 1 topoisomerases never require an ATP molecule. Type 2 topoisomerases always require an ATP molecule. A type 2 topoisomerase is DNA gyrase.
Prokaryotes have a special type 2 topoisomerase called DNA gyrase. DNA gyrase decreases linking numbers in steps of 2.
Decreases linking number. What do you mean by decreases linking number? That means it is going to introduce negative supercoiling. And type 1 topoisomerase was introducing positive supercoiling. It was introducing positive supercoiling. Introduce positive supercoiling. Coiling. And in the other one, it was introducing negative supercoiling. Introduces negative supercoiling. Any doubt? Moving forward.
DNA gyrase is responsible for the negative supercoiling of chromosomes in prokaryotes.
Okay, it is responsible for the negative supercoiling of the chromosomes in prokaryotes.
You have discovered.
Now, this is about the apply your knowledge test that is referred to as the questions, the MCQ questions that are going to be asked about the particular topic. So is the topic clear to you or not? So that's the most important thing. So
Prokaryotes.
I'm going to end up here only. This is all about ifs and buts about the twist and writh that are being associated with the DNA molecule. So what are these twist and writh? We have looked forward and would be meeting you soon